I introduce very shortly Professor Cozzani, is Vice President of Accademia, and he will speak very shortly about the reason of Accademia and many other things. Please.
Thanks, Salmo. Ladies and gentlemen, good morning. I speak a little English because I prefer che Almo fa a translation fra Italian language ed English language. Ieri ho apprezzato molto i vostri lavori, veramente molto interessanti, e spero che anche oggi sia altrettanto. Nel pomeriggio voi andrete nel Parco Nazionale delle Cinque Terre, ospiti della mia amica presidente del parco. Il Parco delle Cinque Terre è un territorio molto delicato, fragile, e un turismo così massiccio può dare gravi danni al lavoro dell'uomo fatto in millenni. Oggi, in 50 anni, 100 anni, è possibile distruggere tutto. Yesterday, nella relazione del professor Almo Farina, ho visto un quadro painter di Telemaco Signorini, un noto pittore italiano a cavallo fra l'Ottocento e il Novecento. Ed era un quadro di un luogo di Rio Maggiore, the first of the Cinque Terre, la prima delle Cinque Terre. Ebbene, in quel quadro c'erano 3 persone. Oggi, nello stesso spazio, nello stesso luogo, ci sarebbero almeno centinaia di persone. C'è una trasformazione violenta del paesaggio che è deleterio sia per gli abitanti, gli inhabitants, sia per il paesaggio stesso. E quindi sta a noi tentare di salvaguardare questo patrimonio e di regolare al meglio un flusso turistico veramente eccessivo. Buon lavoro.
Thank you very much. I appreciate these warm words for us, for the code biology. It's really important to recognize that our theories, our ideas can be spent really in the landscape, in the social context. This is another mission that we have. So today we will visit the Cinque Terre. It's a very fragile area with a mass tourism where people is coming from everywhere from the world, of course. But because tourism is a special innate code, in my opinion, ecological code. And we are studying the codes, and we could modify some behavior in the code making. And I stop because now it's time for my colleague. Thank you very much. Today we have more occasion to speak about this concept at Cinque Terre Park. Another very short communication before the session. It's not possible to use the boat. The sea conditions are really hard. So we move by train 15 minutes. 15 minutes we are in Manarola. So it's more comfortable, but not so beautiful as the navigation. I'm sorry. I asked, but nothing happened. Okay, it's your time, Lutz. Thank you.
Okay, good morning, everybody. So before I start my talk, I would like to thank Almo for organizing this nice conference in this beautiful place. And it's always, I'm always glad to be at one of the code biology conferences and to meet some friends again. And I'm very much looking forward to the excursion today, of course. Yeah. Okay, now it's time to talk about more mathematical codes, and the topic will be the spider web of error detecting codes in the genetic information, which is joint work with Elena. And it's most of you probably have heard some of our talks before, so the background on circular codes. This is the name of the error detecting codes I'm talking about, but maybe or perhaps some of you haven't been there. So what I'm trying to do is to give some introduction, which will also be the introduction for Elena's talk later on. So maybe I'm going to take a few minutes more, and then she's going to take a few minutes less then. And after that, I will tell you a little bit about these creatures, the circular codes that have been found actually in nature, and then some recent results about the structure and the properties of these codes. Okay, so here's the biological background that perhaps all of you know. The genetic information is given in this in the DNA, as we know, which has a double helix structure. So there are two strands that are tied together by complementary pairing of the genetic basis, the chemical basis. And usually A and T pair and G and C, and then they build hydrogen bonds, and some of them are stronger than the others, and that's why we have this double helix structure. Okay, now in the translational process, the genetic information is first copied to this RNA, and then it's translated into the sequence is translated into a sequence of amino acids, which then folds three-dimensionally to a functional protein if everything goes well. And this translational process is based on the genetic code, which is a proper code in Marcello's sense, I think, because it, I hope, is correct. Yeah? It connects in an arbitrary way these triplets of bases to the amino acids, and as we know by experimental proof, this can be modified. And it is redundant, as we can see, so some of the codons, these triplets code for the same amino acid. So we have amino acids that have a degeneracy of degree 1, 2, 3, 4, and 6. And then this process is very fast, and of course, therefore, it's quite error prone, and there can be errors. Of different kinds. So the first one is of course point mutation, which does not really have a, well, it doesn't have a proper effect, but from a theoretical point of view, it's not so serious because it only changes one of the codons here, so replace U by, let's say, T. So it will only affect one of the amino acids in the translational process. But you can also have insertion or deletion, which means a basis is added in the genetic information or it is deleted, and that will result into a frame shift of the whole sequence, which might also appear just because the ribosome skips a basis, one or two bases. So these errors that might appear will have a serious effect on the corresponding sequence of amino acids, which can then look completely different. Okay, so given such a sequence, we have actually three possibilities to read it with respect to the corresponding frames. the correct reading frame that is initiated by the starting codon. So this is called frame 0. And then a shift by 1 basis. So instead of ATG, the first codon that you read is TGA. So you have a shift of, you skip the first A. Or you can skip the first 2 letters, and that will result in frame 2, sorry. So you start by reading GAC. If you skip 3 bases, then you are again back in the correct reading frame. Yeah. And here's an example where you can see this sequence results in this polypeptide, and then if you skip the first one, you see that immediately the process would stop because you obtain a stop codon. Okay, so there should be mechanisms in nature that prevent these frame shifts, or at least detect frame shifts, maybe correct them. One option would be that in frame 1 and frame 2, you will have more often stop codons so that the process terminates and not energy and resources are not wasted, and this is actually the case. And another mechanism could be circular codes, which are a weaker form of comma-free codes that were already suggested by Crick for this frame shift problem. Okay, so I'll give you the definition of circular code in a moment, but basically it means that a circular code can detect a frame shift in the sequence. Okay, so how were these creatures found in nature? There was a huge statistical investigation done by Arquez and Michel in 1996. So they considered genetic data of all the kingdoms using large databases, and they identified 3 sets of codons, one in frame 0, one in frame 1, and the last one in frame 2. As you can see, they had different sizes, and they contained those periodic codons because they are mentioned here because they play a special role in the theory of circular codes. So how did they identify these 3 sets of codons? And that's the important point. What they did is they calculated the codon usage of the codons in all this genetic material, and then selected those codons that had the highest preference for frame 0, but in comparison to frame 1 and frame 2. So they did not just look at the codon usage, they didn't take the codons that had the highest codon usage in frame 0, but only with respect to the other frames, which means, for instance, that this AAC is assigned to frame 0, while ATA is assigned to frame 1, because here ATA has the highest frequency here compared to frame 0 and frame 2. However, if you, let me find an example. Yes, for instance, the ATG has a higher codon usage in the correct reading frame than AAC, but it's not assigned to frame 0, but it is assigned to, let's see here, it's assigned to frame 1. Okay, so that's the important point. It's not just the codon usage, but with respect to the 3 frames. Okay, so that was a purely statistical investigation. No proper theory behind this. But then it turned out that this set that was found in frame 0, that was assigned to frame 0, excluding the periodic codons, and that was for some theoretical reason then. But if you exclude I think it was the 2 periodic codons that had the highest affinity to frame 0, then this set, as I will show you, has some nice theoretical properties from the coding theory point of view. Okay, so these are 20 codons, and unfortunately they were only coding for 12 amino acids. There were high expectations that luckily they would code for all the amino acids, but they didn't. Okay, so that's the set that was found by statistical investigations, and then a lot of research was done, especially by Christian Michel, and he could show that this set, which we call a code now, has a high frequency or appears significantly often, for instance, in the small and also large subunit of the ribosome. So there is some hints that this set of codons has a proper function in nature. Okay, so what is the nice property that then turned out to be true for this set, and actually for the three of them, also the ones that were assigned to frame 1 and 2. They are what we call circular codes, and the circular code is one that if you take these codons and you concatenate whatever you want, but just using the codons from this set, then this concatenation has only one decomposition when you put it on the circle, which means that you connect the end with the beginning. So you read the finite sequence and you continue reading it, repeating it. Yeah? This is putting it on the circle. And whenever you do this for any concatenation of codons from this set, then the decomposition into words from this set is unique. So there is no other, which means that if you have a frame shift in this sequence, you will eventually detect it. Okay, so here's an example. This code, which has only 6 codons, so in our sense, a code is just a subset of the set of codons, is not circular, because if you concatenate these 3 codons and you take a frame shift of 2 positions, so you start reading here, then GTA, GGC, and also TAC are codons which are again in this set. Okay, so if you repeat this short sequence again and again, and you have a frame shift of 2 positions, you will not detect it, yeah, because the shifted version is still a concatenation of words from this little set. Okay, so it's impossible to detect it in that sense. However, if you look at this one, this set here, then it's circular, because whenever you concatenate these codons, and this is just an example, you have to test all the possible concatenations, of course, but if you take this concatenation of 3 codons from this set, you have a frame shift of 2 positions, then you see the first codon that you read in frame 2 is still in this set, but then the next one is not. So you'll find that there was a frame shift. Okay, this is what we call a circular code. And then a stronger version is the comma-free codes. I'm not going to talk about this, but Elena is going to use it later on. Comma-free means that you detect such frame shifts not eventually, so after some time, but immediately. So if you take a concatenation, you do a frame shift by 1 or 2 position, then the first codon that you read is immediately out of your set. Okay, so it's a stronger condition, and Crick was hoping that that nature was only using such a comma-free code, which then turned out to be wrong. Okay, so this is circularity, and then there is more properties which I will just shortly present to you, namely the C3 property and the self-complementarity. Self-complementarity is quite clear. It means that if you take a set of codons, it's self-complementary if with any codon there is also the anti-codon inside, so the one that sits on the opposite of the DNA. So that means self-complementarity. In mathematical terms, it can be written like this, but it's not important for our talk. And the C3 property means basically that with a set of codons that is circular, also the 2 shifted versions are circular. So in our sense, the 3 sets that were found statistically by Arquez and Michel, all of them are circular codes. Okay. Sorry. So this code that was assigned to frame 0 by Arquez and Michel, purely statistically using real sequences from nature, has these nice properties. It's a self-complementary C3 code. So all the shifted versions and the code itself is circular, so has the property of detecting a frame shift in each frames. And that's why it became, of course, interesting to study those creatures, and it was likely that this could not be a coincidence. But maybe this this code, either it is still be used in nature, or it is a reminiscent of some earlier mechanism that was using circular codes. Okay, and Michel repeated the statistical analysis recently with a much larger data set, and it gave the same result, actually. Okay, so here's just a list. By computer calculations, you can calculate how many codes of the various kinds there are, and of course it's important to mention that there are only 216 subsets of the set of codons that have the same properties as the one that was found in nature. So they are maximal, which means 20 elements, 20 codons, self-complementary, and these three property. So it's very little. As you can see, there are many more. Just if you skip this one and this condition, there are many more possibilities, of course. Yeah? You have to take 20 codons out of 64, which is a huge number. But only 216 of those possibilities have this nice property of the natural one. Okay, and for the comma-free codes, there are 408. So it was interesting to start these 216 codes, and we have two central questions that, when you're a mathematician, are straightforward. You try to see what is are the essential properties of these codes and how can they be constructed. And this, Elena is going to tell you a little bit about this. And the next one is, since we are also in code biology, we will ask how are these codes related, and can we somehow use them to explain some biological phenomena. Yeah. Okay, and this is what I'm going to present you. And what we showed recently is this spider web of circular codes. So I'm going to explain why it is of this form. But what we could prove is that the 216 circular codes that we have can be arranged in this form as a sort of spider net that has a cut here. Okay, this is unfortunately not finished, the spider web, yeah. So there is a cut. And each edge here means that you can pass from one code to the other. So the vertices are the 216 codes. And the edge means that you can pass from this code to this code and from this code to this code by some mechanism. Okay? And this is what I'm going to present you. Going from the outer circle to the inner circle was known before. Namely, we could use group theory, and that was work with Diego Gonzalez and Simone Giannarini. We could use some group theory and apply transformations on the alphabet. So exchanging the chemical basis bijectively by itself. So let's say replace all the A by T or the G by C and so on. And there are 8 such transformations that form a group, and that then classify the 216 maximal self-complementary circular codes into 27 what we call equivalence classes, each of size 8. Sorry. And this is these are the transformations. Of course, they contain the identity. And passing from the outer circle to the inner circle means that we apply one of those 8 transformations successively. Okay, that's why we have 8 circles. So that was known before, but then we never studied what is the relation between the equivalence classes, the 27 equivalence classes. And this is what we did recently. Before I'm going to show you what the relation is, let me mention, because then we can come back to the biology. The group theory already proved that within such an equivalence class, so these are just is one of the equivalence classes, 8 of the codes, the numbers do not play a proper role. You always have what we call the winner code and the loser code, which means one of the codes perform best with respect to codon usage independently of the species. And this was a nice result, because what Arques and Michel did in the beginning, he looked at all the species together, all the genetic material. And they found this code X. But if you look at special species, then some of the other 216 codes perform better than the one that was found by Arques and Michel. Just in the average, it was the best one. Okay, so there might be a relation between the species and the maximal self-complementary circular code, C3 code, that it is using. Okay, and the group theory already said, okay, if you just look at one of the equivalence classes, then it's independent of the species. So in any species, this code number 173 performs better than all the others in that equivalence class. But in another equivalence class, there might be another one that is doing better. Okay, so that was known before. But as I said, between the equivalence classes, we did not have any good insight. Yeah, and so what we did is we now defined a relation on the equivalence class, or first of all on the codes. We said that two codes are close if they have an intersection that is as large as possible, namely 18 codons. So there's just a pair of codon-anticodon that has to be replaced by another pair of codon and anticodon, and then you pass from one code to the next one. So they have a large intersection, and there's only little modification between those two codes. Okay, and this is an equivalence relation, and it gives you a graph, namely by connecting. You take as vertices the 216 codes. You connect two vertices if the intersection is big, has size 18. And this is a connected graph. It has the 216 vertices, some edges, and then you can start studying this from a graph theoretical point of view. So these are just some properties that we mentioned, but it's not the essential point. Okay, this is how it looks like, the graph. And one theorem that we could prove is, and that we didn't expect that, and it was quite difficult to prove it then in the end. Of course, computer calculations can show it to you, but then the mathematicians always want to have a theoretical proof. This graph is bipartite, so it can be colored by two colors, okay? And then neighboring vertices never have the same color, which is a nice result, and as I say, we didn't expect that. So here we have some more theoretical results that we proved, but I'm not going to present all of this. What we did then is this equivalence relation on the 216 codes induces one on the equivalence classes, okay? Namely by saying that if you take one of the equivalence classes and another one, you connect them if there are codes inside, so that in this old relation they have an intersection of size 18. You have to prove it's compatible with the group theory and so on. But this gives you another graph that displays the 27 equivalence classes. Okay, and again, there are some graph theoretical properties that are not true for the graph, but as we can see, so this is much more visible now because it has only 27 edges. For instance, it can be colored with four colors now. And as we can see, it has two loops here, which means that within an equivalence class, you have two codes that are very close together, equivalence class under this group theory. But the most important point, and that brings us back to this spider web, is that in this graph, which is not Hamilton, but you can find a Hamilton parse of maximal length. So you can go through the parse, through the graph of those equivalence classes, and the parse, the connected parse, has length 27. So you cover all the equivalence classes. And this corresponds to one of the circles in the spider web, because it means that by, for instance, there is one way here, yeah, through this graph G27, take the equivalence class of 173, then you pass to 98 and so on, and you end up in 120. So these are all the 27 equivalence classes, and you pass from one to the next one just by changing one pair of codon-anticodon. Okay, and this gives you this spider web. So passing from the outer circle to the inner circle means you apply group theory, and then on the circles, you do this replacement by a pair of codon anticodon. And of course, there is a way of describing all the 216 codes, because you can just start with any of them, go to the proper circle, and then do this process of replacement, and you find any of the other maximal self-complementary C3 codes. And now the idea is to use this and to see, does it give us some phylogenetic information? Yeah, so maybe species that are using this code the most, yeah, are they closely related to species that are using this code the most? So that is something we still have to investigate, but it could be an idea that species are close if they are using the C3 codes that are close in this spider web. Yeah. Unfortunately, it's not connected here. Yeah, of course, so the last and the first code on one of those circles has a smaller intersection, not 18, but I think sometimes it's 14, which is the minimum. Yeah. Okay, so that is the recent result. As I said, the biological implication is still work to do, but that's what I wanted to present, that we have now some kind of understanding of these circular codes, and yeah, so this is probably material for the next talk at the next conference. Thank you.
6 minutes for discussion.
Just a short comment on self-complementarity, which is known for decades, almost 1990s. That property is also strongly connected to ligand binding. So all antibody receptor interactions can be done, and that self-complementarity has been used from 80s in the so-called molecular recognition theory. So basically, inside the algorithm of Micheli is also embedded in that self-complementarity ligand receptor interaction, and it has been shown for more than 50 ligand receptor systems. So basically, within the circular code, we have a sort of circular pattern of ligand receptor interactions, which is now, during the last 15 years, extensively used in pharmacology. So it's even more important than just finding a circular code. And another thing, of course, is I will present that tomorrow. We did some relational, I would say, decomposition using Kleinian groups of all genetic codes, all 33. And there are some regularities which point out that the web the spider web you're representing had some exclusions. I don't know the consequences yet because it's all new, but I assume that there are points within all possible codes that are automatically excluded from mutation and from the standard genetic code and the rest of 32 codes. So I think that combining your part and our part, we could even speed up and tracking the possible sequences, circular codes, and exclude some of which are simply not present in all relational tables. I don't know how it will look when we put Kleinian operators in the web on the nodes, and made all the exclusions. But it might look very interesting. We did some research which is in progress, and there is a very strong, actually, proof that that it should work for evolutionary trees, so for tracking the segments of evolution. But we have to work some more on that topic. Thank you.
Especially what we are looking for.
Other question? Okay. So thank you very much. You speak here or what? Yeah.
Moment, I'm looking for the presentation. Where is it? Could you please? I will continue. Thankfully, Lutz has already provided the necessary definitions, so he did the hardest part of the work. So I will just continue, and I will try to show, we try to understand, as Lutz already said, how these codes, 216, in particular, are constructed, construction principles of this codes. And we came closer to the answer to this question when having some tool which I will present here. How it works here? Yes. No. Excuse me. Then I will skip the definitions because Lutz already did it, and just remind you briefly what what classes of codes are we speaking about, and we will speak mostly about the so-called comma-free or C3 codes. Self-complementary C3 codes. And here are once more the necessary definitions of the C3 codes. I remind you that a code is called a C3 code if the code itself, as well as it cyclically shifted codes, are circular. And then self-complementary, as already said, that the code contains, together with each codon, its anticodon. Then here is a small reminder. Lutz already told about this shortly, but I will say it once more. If we have such a code, such a code class. Let us say comma-free codes or circular codes or C3 codes. All these properties are preserved when applying one of the permutations of the basis, or if we reverse each codon of the code. But the property of the self-complementarity is only preserved when we apply the 8 transformations we indicated before. It will be seen, you will see it in the tool, it can be visually seen there. So then I will present now some tool programmed by a former student, of ours, and the tool is good for establishing some hypotheses, which we later proved mathematically, of course, because a computer proof is not a real proof for a mathematician. Then let me show you the tool itself. Here it works. So you see, first of all, we have here 60 codons. Why 60? Because, as Lutz already mentioned, the periodical codons cannot be a part of such a error-detecting code, so we excluded them. Then we ordered the 60 codons in the way that we have a codon and its anticodon nearby. And in the same row, we have shifted versions of this. Let's say these two and these two are shifted versions of each other. Means visually that only one or at most one pair from each row can be in such a error detecting code. Then we can I think I will sit better because I have to show the tool and this microphone in the hand. It's not so easy. Then I have to push this now. Then we can choose options here. For example, which class of codes can we or do we like to explore? It's comma-free or circular or C3. And each time this table of numbers on the right side are the numbers, the lists can be downloaded here, but each number stays for some maximal code of this class. For example, if we have chosen this C3 option, then we have 216 numbers here, and each number and each row is here for such equivalence class, as Lutz said. It's just a visualization. So then we can also choose the option self-complementary or not. We can choose here obey or ignore, so we can change this, and we can also choose the option maximal yes or no. You will see in one example I will show you that how we applied this option. And okay, then we have chosen now C3, obey and ignore. We don't necessarily want to have a maximal code. Maximal means that we have 20 codons, means from each of these row we have 2 codons. For example, if it is self-complementary, we have 1 codon pair. Some years ago, we already proved that this, you see here, that codons are colored in different colors. So we divided additionally this set of codons into classes, color numbered, and it is class 0, 1, 2, 3, 4, and 5. So we say, have in general 6 classes here, and it means how did we selected these classes. It depends on the construction of the codons of the class. Let us say codons of the class consist of contain 3 different letters, or just 2. It cannot be just one, because we excluded the periodic codons. For example, let us take a look at the class with the number 0. You see here we have codons ATA and TAT, and C, GC, GCG, means that this class contains of codons which are symmetrically, means the same letter is on the first and third position. In the middle is another letter, and these 2 letters or nucleotides are connected by the complementarity, A and T, C and G, for example, like this. Or take a look at the class 3. It will play a role later. Here we see codons which contain of 3 different letters. For example, CTA, TAG, and so on and so on. And 2 of these letters are connected by complementarity, and it is also position in this. Why have we decided to take such a division of the set of codons? It explains if we begin to choose different codons from these classes. For example, if we take this pair, we decided to choose the option self-complementary. It means if I click on this, I choose immediately 2 codon and anticodon. And you see we have this in yellow, so we have chosen this this pair, and we have some pairs which are now in purple. It means these pairs cannot be in the same code with this yellow one. And as I said before, if we haven't chosen anything, you see that 1 or 2 pairs are already excluded. And it was one of our first results several years ago that these pairs cannot be a part of a C3 self-complementary code. It is not possible. Actually, a circular self-complementary code. And so by clicking of these pairs, we established some hypotheses which codons don't like each other, and so we formulated it as theorems for all classes of codes. So like for comma-free code, codes for circular, self-complementary, and for C3 self-complementary codes. 3 kinds of theorems. I will not bother you by explaining these results in detail, but I think you understood what is the idea. So we described these codons in a general way, and so we put the results. If the pair is in the code, then the pairs like this cannot be in the code. So what gives us, what is the reason, why is it interesting at all? Because it gives us some insights into the construction principles of such codes. For example, one question we could answer by means of this tool. First find an example, and then of course prove theoretically. it was not known that if you have already such a C3 self-complementary code, not maximal, but some, let us say, 10 codons. Can you always extend this code up to a maximal one? It was, for example, an unknown question. And we answered this question negatively. It is not always possible. I will show you an example. So I wrote here some pairs of, you have, the idea is that you have to choose the pairs the way that you can completely exclude at least one row in this table. If you succeeded in this, so you have already a code which is C3, self-complementary, but cannot be extended anymore. So, and in this case, we have to choose, again, the pair TAATTA, then GCC, GC. It's already two first rows are served. Then ACT, the next one. You see, if I choose this one, I exclude already many codon pairs. Then CAA, this one, and AGG. And now you see, you can ask me, you can still choose this T, C, A, T, G, A. But taking a look at the list of the maximal self-complementary C3 codes, you see that all of them are excluded. And if I now choose the option obey the maximality, you see that I cannot choose any pair anymore. So it is the way you can imagine to construct such an example. Just manually, it is almost impossible. The number of combinations are so huge that only by means of such a tool, it is not a proof mathematically seen, but you obtain some hypothesis and you can look further. So this was the first interesting result we obtained with this tool. And the second one I will show you now. We were always curious about the number of the maximal self-complementary C3 codes, 216. It's such a nice number. It's 6 to the power of 3, for example. And so we thought it cannot be by chance. And now with this tool, we could come closer to the construction principles and explain this number. And the main idea, I will show you only the idea of this. We take a look closely at this class with the number, it's 0, 1, 2, 3. Actually, as you can see, this class contains of 8 codon pairs. It's not by chance. This is the number of the elements of in this L group. All these codon pairs can be mapped into each other by means of these transformations. Actually, if you find one example, you can find another 7 by mapping just by using these maps. So, and the observation is in a maximal self-complementary C3 code cannot be, it cannot contain 4 pairs from this class. Only 1, 2, or 3. Because if you take 4, it is immediately not circular. So we proved this. And if we consider now the situation, we have chosen 1 pair from this class. Means I cannot choose this one, and this one, because it should be only 1 from this class. means I have to choose from the rows the elements of on the class number 2. And you see, if we have chosen these 4 pairs, we have here no option, here no option, here no option, here no option, and here we have 2 and here 2, 2 options to choose. And this is the way how we have shown that this number 216 appears. Means, for example, in this case, we can we have for the choice of the pair from the class 3, 8 options, and then in every case we have 4 more options, means 32 of this kind. Then if we choose 2 different codon pairs from this class, we have another, or we have described this in our paper, so you can read this in detail. The paper is submitted, but not published yet. No? But we hope soon. And this is actually all I wanted to show you today. Thank you for your attention. Please ask questions.
Time for discussion. Hour?
But questions first.
Sorry, I just started immediately thinking of, let's say, codes in social communication which have this mechanism. And it's interesting, so in human language, if we, it's quite possible that, so not every single language has breaks between the words. So for example, if you read hieroglyphs from top to the bottom, they're not subdivided into words, and sometimes it's, well, Japanese friends can't always say where one word ends and the other begins. It's not essential. But in languages we use in our practice, there are such mechanisms. At least when I was listening to both of you, I came up with one. So in English language, we have the combination of letters Q and U, yeah? Which seems to be extremely redundant. So basically, if language would if human language would develop always according to the TIF hypothesis of efficiency, this U would be dropped. But no, it's not redundant, because if we break the words, let's say the frame shifts in human language, let's say 2, 3, 1 or 2 letters to the right, yeah? Then you have a break between Q and U, yeah? And if there would be a break somewhere else, you could still say, okay, I don't know all the words of the language, but this is something I didn't know, because there are no general rules that preclude creating the words according to these combinatorial rules. But if the break occurs here between Q and U, then it's clear that there is some error in it, because the rules of English language do not allow you to put Q separately from U. Yeah, so on the left side you would detect this error. Maybe that's as close as you can get to, you know, to this circular code.
Zip distribution, and I know that there are some works in this area. For example, Omar Paredes, whom we know, he applies such techniques, but it is not about circularity in our case. So what I am speaking about is probably that you have some unbreakable combinations of letters. It is not the case in because in the genetic code, because we have only words of the length 3, and we use all of them. No?
case of it would be an extended case of quantitative correspondence analysis, but on a wider scale. But you also, finally, in the end, you got tabulated operators, which by Boolean functions and two-digit letters represent each symbol. And the symbols, you have amino acids, and the codes with information carried out by third functions are codons. So we are working on the tabulated positions within and break it into relational tables, which are joined by a single column, and which have a key between the first and third base, which is primary key, and then you have it's an extension of quantitative correspondence analysis.
Thank you very much for your talk. Very interesting. I actually found the program your student developed really fascinating. But I just have just curiosity. When you put obey and ignore, when you put obey, the results came up immediately. This is the case always, or sometimes it actually takes more than? Yes, the combination. Immediate. All the okay. Mm.
You see, for example, here in the list of the maximal codes, you see these codes with number 80, 85, 131, and 157. They contain this combination of codons. You can see immediately which codes contain this combination. And if you exclude more and more, you see that this table on the right will contain more and more purple places. So it's visually.
Other questions, comments? Okay. So thank you, Ellen.
Thank you. So there's just one quick announcement that we would like to make. Actually, I have some slides. I don't know how to. Okay, can you help me with the? Yeah. So as you know, Robert Prince couldn't come to this conference due to time reasons, but he sends his regards. And Elena has already spoken about this. So she mentioned yesterday that there is some project that is going on with Robert. And Marcello also told us that he is the code club. No, this one. Yeah, the one down. Yes, thank you. There is some project going on that Robert initiated. He was searching the literature for all kinds of codes. So just basically was looking for the name code when it was mentioned, and he created a list of papers that contained code in some sense, and this was the idea, or the initiating idea that came up to try to build a database that contains all the information about all possible codes, which is a big task and a huge work, and then try to classify the codes by their characteristic properties, so that we can see similarities between ecological codes and circular codes and so on. Yeah, and then that will probably help people to work together and to better understand them. So the idea was to create this code club, and there is a little project that where also Candice, Anna, Adam, Omar Paredes are involved, and it's funded by the German Research Foundation, and it's the starting point for this work. So Robert, who is the main proposer, suggested to build this code club, and his idea was to, in order to get this data, up and down, no? Let's see. Yeah, this works. Okay, so the idea was, in order to collect this data, to offer a seminar, open access seminar by Zoom, more or less every month. So the idea was to meet for 1 or 2 hours and to invite, so it will be open to anyone, but to invite some people to talk about the kind of codes they are working with, to give some information, so that we can collect the data that then will be put in this database. It's at the very preliminary stage, and the idea was just to announce it here, so that you are aware of this project. He had some idea, for instance, this could be a possible schedule for November, and then there is more in December and so on. But it's not fixed yet. It's just an idea how this could work, so that we talk about this kind of codes in the first meeting. Someone presents information about this, and we're trying to extract the data that we need. There will also be a web page. If the guy can come again and change the PDF, I can show you. We also have a questionnaire, so that is, of course, the best way to yeah. You can just close this, please, and then there is another one which is called Code Program. Up, yeah, the 2, 3 up. This one, yes. So we have also created an initiatory questionnaire that people can fill in and answer questions about their codes. And we have some copies here. Maybe we can produce a bit more. The idea would be that we ask you to take a look at this questionnaire. Maybe you can already fill it in, but basically to see, does it fit your understanding of code, or what you're working with? Should we ask other questions? Are these the right questions in order to provide all the information and so on? So we have some handouts here, and I can't read it. I think it's here. This will be the webpage, biologicalcodes.de. But we're going to send this by email to all of you. It's just a first announcement that we would like to make, and it would be very nice if you could take a look at this questionnaire to give us some feedback, perhaps by the end of Friday, so that we can collect it and have some ideas of how we can improve this, or how we can create this questionnaire. All right, thank you very much.
Okay, thank you. This we are at the end of the first session. Now we have a coffee break. And we have a small change of the program. We have a presentation from Branko Dragovic at the same time. And in the place of Garcia Castro, we have the presentation of Mikhail Hilin. Okay, so now we have time to move to the coffee break in the hotel. And we can wait because they are waiting for, we are in advance. Marcello, have you some comments about this? No, it's fine. Okay. The people that are in this hall will participate to the field excursion, or there are some people that they can. Quando? Per cosa? alla solita invece fra mezz'ora, alle, no, alle 11, sì, 11. Sì, sì, it's the same, nothing is changed.
Devo dirvi che per oggi,
Sì.
se prendiamo il treno delle 2 e delle 14:15, bisogna che si trovino, come facciamo? Li facciamo trovare qua all'1:30 andiamo alla stazione?
E decidiamo dopo, quando ci abbiamo più persone.
Vabbè, ma ormai
as we have a very small change of the program, but we have 3 presenters. At the end of the presentation, I hope to distribute you the ticket of the Plan B. Plan B, moving to the Cinque Terre by train. So Olympia, our fantastic assistant, is at the railway station for buying the tickets. So she will come back, I hope, or will depart with the money, and then I explain shortly how to arrive. You know where is the railway station, no? And the meeting is under the big tree, Platanus orientalis, of the railway station. And the Manarola is the second stop of the local train. First is Rio Maggiore. No, it's not for you. The second is Manarola, is for us. And I recommend you to have attention to your personal effects, because there is a lot of tourists and a lot of thieves. I presume. So, no other recommendation now, but the time is a quarter to 2. A quarter to 2. Before 2. No, at the railway station. Yeah, you move where you like for lunch, and then the meeting point is at the railway station with your ticket. Your ticket will be distributed now. So you are free to move, and the train is a quarter past 2, but I appreciate to stay half an hour together to organize ourselves, if you like. Otherwise, the train is a local train for Manarola, Levanto, and the second stop from La Spezia. It's okay, more or less. I try to be present in any moment of your life, but it's not possible to accomplish everything. It's in front. There is a big tree. Under the big tree. Okay? And I will cry, code biology, and you will come, okay? Thank you very much. I'm a little joke, I apologize. Well, Branco, it's your time, Professor.
Yes. I will.
Okay?
Okay, good morning to everyone. This is 10th International Conference in Code Biology. I was participated at 2 of them, and this is third for me. I was in Granada, and I was also in Germany near to Frankfurt. I think that this series of conferences was very successful, and this is also Jubilee. And first of all, this is Jubilee for successful these conferences, and especially for our founder of this code biology, Marcello. And I wish also in the future to have
this better and better, to Marcello, to Elena, to Lutz, and other members of the Garmin board. As we see, here we are from the different fields of research. And my background is in theoretical and mathematical physics. And I had research in several parts of theoretical mathematical physics, from string theory to the cosmology. I started to interest in code biology, in the modeling of the genetic code about some 18 years ago. It was at the beginning to apply some formalism, which I knew from the mathematics, to the modeling of the genetic code, especially about similarity between codons. Today I will speak, first of all, about physical information and biological information. Start with physical, of course, at the beginning, some small introduction. And I will not have time probably to speak about this p-adic modeling of the genetic code. It was presented 2 times earlier at this conference and many other places, and there are also papers and so on, and this is rather technical also and so on. So, this is here. Okay. At the beginning, what is information? Intuitively, we distinguish between what is and what is not information. But to give definition of information, it's the question. According to the recent developments in sciences, information is recognized as a fundamental concept, like space, like time, and like matter. And these 4 fundamental concepts are interconnected, substantially interconnected. Since information is a fundamental concept, we cannot give a definition, because when we are giving definition, we should use more fundamental, more elementary concepts. Yeah? And since this is so fundamental, there is no complete definition of information, but there are definitions in different fields, depending on what people do. Here I will use working definition for these aspects. And for me here, information is a special state of matter with some meaning. And the information, on information depends evolution of the universe and also evolution of some parts of the universe, like in biology, in the genetic code. So depending on the complexity of the system, or in general, one can speak about physical, about biological, and other kinds of information. So I will now speak about physical and then about biological information. So when we want to speak about physical information, as I said, that information is related to the special state of the matter. In fact, in general, when you have, in principle, many possibilities, but nature take just one or some, then it is information in physical sense. First of all, I would like to recall that there are fundamental physical constants, like speed of light in vacuum, also the Planck constant, the universal gravitational constant. They are three mainly the most important. There are also many others, yeah. Fundamental physical constants. You see, they are given from the beginning. They are related to space, time, and matter. That's one point of view. The other one, if you look at the elementary particles, they have some properties, let us say for electron, for proton, and so on. They have some properties like mass, charge, spin, and they also have definite values. So, in principle, electron or any other elementary particle could have any other value of the mass or the charge. But you see, from the beginning, it is given just definite value. And these definite values for fundamental physical constants and for the properties of elementary particles, they are given from the beginning of the universe. And they are constant. There are no change of these properties, these values. So there are some invariants during universe evolution. If you want, you can ask questions during also my talk. Here, okay. For example, in physics, you know it also, what does it mean special, for example? Let us take some very simple example. Let it be related to the motion of a free particle. It is given by this very simple differential equation of the second order. The solution of this equation is this one, x is equal v, which is velocity, times t plus x0, some position. And this v, velocity and position, they define state. if we don't take special values of this v and x, we have infinitely many possibilities. But for some concrete case, these integration constants, we have to take some definite values. And then some special motion of a free particle is defined when we take these values for v and x. So I want to say that we have here also the notion of state, and we have for the concrete case to take concrete value of the state. Also, in the case of the general relativity, which is by which one can consider evolution of the universe, there are here two equations, which are so-called Friedmann equations, and there is this A here, which is some scale distance, and you see, to solve this equation here, which is related to the evolution of the space during time, and it depends on the matter. You see, to solve this, we have to know also, we have to precise state, where the state is connection between pressure of the matter and this rho, which is density, energy density. So you see that when we concrete some special cases, that we always have also some work with the state. We should know the state. Okay. Now, let me start with the biological information. As you know, information plays one of the central roles in biology. And like Marcello liked to say, maybe not just in this form, but in this sense, biology is chemistry plus information. Here, what is here working definition? According to the definition at the beginning, what is information? Here, biological information or bioinformation is a special state of a biological system with a meaning. Here, as an example, biological system can be DNA or RNA. And in fact, DNA and RNA are main carrier of the biological information. I would also like to point out that there is no information without carrier. There is always some physical system or biological system which is carrier of the information. And mainly biological information is coded by special sequences of the biomolecules. We know it, yeah, very well. So, to demonstrate that we have many possibilities, but realized are only some of them. In such sense, it is information, and when it is also with some meaning. Let alpha be number of letters. For example, molecules that are elements of some sequences. Yeah? And let n be length of such sequence. Then we know that n as a number of words, or number of sequences, is alpha to the power n. And this alpha for DNA and RNA is 4, because we have 4 nucleotides. And for proteins, it is 20, because we have 20 amino acids. So, since human DNA contains about 3 billion base pairs, so it means that there are 4 to this power 3 billions. It is huge, huge, huge. But in the real case, yeah, how many humans there are? Now, something about 80 million. But this is, you see, a very, very huge number. So, between many, many these possibilities, in biology, we have only some special, yeah, and these special are with some meaning. Also, if we, if we want to compare this number here with the estimated number of how many hydrogen atoms there are in observable part of the universe, it's about 10 to 86. But if you compare this, it's much bigger. Also, if we look at the proteins, let us say that average length is about 300 amino acids. Then you have 20 to 300 possibilities. But in human body, there are about some more than 100 different proteins. So here you see there is a big difference between possibilities and what we have in our body. DNA, as we know, contains genes, which are information, in fact, for the proteins. There is also what is called junk, but I don't think that it is junk. Some of this is related to the regulatory information. Maybe in the future we will know more about it. And I think that it should be more and more related to the regulatory information. So regulatory information is related to proteins, in some part to the non-coding DNA, epigenetic, some external conditions. And what does it mean, regulatory information? In some sense, it is related to when, where, and how much it should be something done. Yeah? And genetic information is a basic illustration of biological information. Protein coding information of DNA is coded by sequences of codons, so you know, yeah, 8. And we know that connection between codons and amino acids is given by the genetic code. Okay, I will just a little bit to say about coding. About p-adic modeling of the genetic code. The genetic code, from the mathematical point of view, is a map from 64, or set of 64 codons onto 20 amino acids plus one stop signal. And how many there are possibilities here? There are, you see, about 10 to 84 possibilities of these connections. But in fact, okay, this change from time to time. There are about, let us say, 30 genetic codes in living organisms, which are also a little bit different. Yeah? And the main question is why the genetic code is such as it is. Yeah, okay, it is a result of the evolution. We cannot say why it is just as it is, but we should find some kind of the model, how to describe it using some mathematics. And there are many approaches, just to mention. Gamow was first, yeah. Crick, Rummer, and many, many others. And in 2006, I started to make modeling of the genetic code using p-adic modeling, which is some part of the ultrametrics. Okay, there are a lot of this here. Just we use, in fact, so-called p-adic distance, which is some kind of the ultrametric distance, to describe to describe similarity between codons. What I have in meaning? What is the idea? If you have, when we have two codons which code the same amino acid, it means that they have the same meaning. In some sense, they are close. When we say close, it means that it is related to some distance. Which distance here? Not Euclidean distance, some other distance. Distance in information space. And in fact, ultrametrics, or in particular p-adic distance, is related to describe this. And it is done very successfully with several papers with me and my collaborators. Okay, since I want to give possibility for questions, I will stop here, just to show also that there are here some list of references. Also, you see Barbieri to reference and many others, Kunin and so on, and some my papers with my collaborators. And here is conclusion. It is just what I said. And thank you for your attention.
Thank you very much. We have 10 minutes for discussion. Questions, please.
You presented information as a fundamental concept of physics, and then said for this reason it's not easy to define because it's fundamental. But then you were also talking about meaning. Okay, I'm coming back to your claim that information is a fundamental concept of physics. Yeah, and that's why it's difficult to define. You know, you cannot reduce it to other concepts. But then you introduced another concept, that of meaning. No, and meaning. Yeah. So it's the same. it's kind of the same meaning. And no, I'm just.
Other questions? Cello? No.
No question that I make my stupid question due to the fact that I don't know your theory. When you measure distance, do you measure it out of chemical properties of the codons, for instance, or out of differences between the proteins that they generate? Is it a distance on the measure that they say input or at the output of the channel, or the communication channel?
You see, you speak about some words in the words of the telecommunication, more or less. Okay. First of all, I start to consider distance between codons, yeah? And one can describe it with this approach. When p-adic distance, you know, when it is the smallest, then codons code the same amino acid. When two codons have the smallest p-adic distance, or ultrametric in more general, then they code the same amino acid. Then one can also use this formalism to consider similarity between amino acids. Yeah? And then also one can consider it as networks, that you have network of the codons and network of the amino acids, and the genetic code in some sense is a connection between these networks.
Yes, please.
Just maybe just wondering about, being coming from the field of linguistics, it's really great to see meaning, which is really for us as a semantic notion, to be as fundamental as at the level of physics, if this is true, of course. But then there's something which is really, just for curiosity, because even in cognitive linguistics, if we want to do something that whether other species than humans can actually have this notion of meaning, we can't really use the word meaning. What we're using is behavior. You could have a stimulus and how is the response to be this. We don't use meaning at this level, which is just one step back, for now to go back all the way to physics. At least I'm thinking of it as action and reaction, just that pure as this. Now to go and call it meaning, I see what you're coming from. It's information part of it is meaning itself. But is it that a long jump? Is it, or it's just wondering, But may have different meanings.
Yeah, meaning with the different meaning.
Last question, because
does your model take any account of the noise occurring during the information transmission? Does your model take any account of the noise occurring during the information transmission?
I didn't speak here about Shannon approach, and as you said that for Shannon it is not important what is meaning, just how many possibilities it has. And in fact he started from the entropy, which is a concept from of the thermodynamics. How many, if you look for the gas, how many there are possibilities, how many states I talked about it. And it can be then related to the information. If you have more entropy, less information. So, okay.
Okay. Thank you very much. Oops. It's time for the second presentation by the colleagues. Michael Healing, please.
Thank you.
You need it?
Yes, sure.
Okay.
Sure, just a second. I think I have to stop it somehow. Please run. Okay. It's this one, the first one. How you made it? Oh yeah. Okay, good. Good. So, dear friends, first of all, I'd like to thank Marcello and Alma for selecting my presentation and giving me a chance to be here among you. And secondly, I'd like to thank both of those people that they adjusted the program in such a way that I'm speaking after the first session where and important things were presented by Lutz and Helena, and well, they represent also a team with missing Robert Prince. And then we had this excellent presentation by Branko. I thank you, because they all constitute some kind of a firm ground for my vague conjectures, I would put it this way. So I'll be speaking about modeling. And to make a link to Branko's presentation straight away, I would like I'm fond of playing with words. You know, by my first education, I'm a philologist. I'm Shakespearean scholar. My first degree was on the last plays of Shakespeare, and I'm expert in literary history, linguistics, all these kind of things, and I'm fond of making tricks with words. So I would suggest that a synonym for my word modeling is another word which you used. It's information. Because form and mode are actually the same things. Latin modus or forma, it's something shaping. So the whole thing about information and the whole thing about modeling is making shapes or structuring something somehow. And here I'm to speak about moments and manifestations. That's something which precedes modeling, which is a basis, which is a starting point. You have some kind of a moment, some kind of an effort, and some kind of manifestation of this method, and you have affordances and outcomes. Affordances as possibilities and some new abilities, and outcomes that come after. And so both are interfaces, because modeling and information, although we are using the word which sounds like a noun, and when something sounds like a noun, it's a thing. That's our immediate imagination. The information is a thing. No, it's a process. And it's a lingual fallacy which we inherited in the process of evolution of human cognition, and in the process of evolution of human communication. We developed some fallacies, some very negative properties which somehow diminish our abilities. And one of those properties is that we take, it's very difficult. One of the things which we can learn from yesterday's presentations, for example, a beautiful presentations by Marcella and some others, that an important thing is this vague, nebulous structures which are constantly changing, which are in the process of change and development. So everything living, weather out there, the biosphere, the starry sky above us, the starry sky is changing. If you remember the Kantian claim about a sky above us, but this sky above us is not a thing. It's something which changes all the time. Every minute, every hour is different. And the moral law inside us is also every moment, every second, it's changing. Everything is changing. So it's terribly difficult to grasp this thing. That's why we rely on attributing momentary states to be things which are fundamental, and processes something which is subsidiary and helping it, which is the other way around, in fact. And that's why you could see this subtitle here. This subtitle could be probably better express my intention. It's our common genesis, or genetical, or whatever you can call it, but formal genesis that integrates everything, and multiple thresholds in this genesis. This genesis is never ending, and it's having no limits, but it has some thresholds, and this would be the outcome of my presentation when I will claim that we don't have any limits. We don't have any strict borders. We have only thresholds, and actually we even have not thresholds, but cascades of thresholds, not a singular threshold, but many thresholds at the same time, combined together. But let me go further, how I do it. Like this. No. Oh, yes. So now I'll tell a few words about myself. So please call me Misha. I like Misha better than Mikhail, which sounds very official. So call me Misha. And I have my own whims, I would call it this way. One of the whims I told you about, playing with words. Another one is that well, I'm motivated for this specific presentation by this grand debate which happened a few years ago about code biologists on the one hand, or only coming code biology. It's emerged in the spirit, and biosemiotics about interpretation and coding, how interpreting and coding are interrelated. And so they were discussing coding without interpretation and interpretation as something absolutely new, novel, unprecedented, which was something like, to my mind, excuse me if it would be offensive, but it sounds to me now, not then, because then I took it very seriously and I was very much involved. But now it sounds to me like a scholastic debate. And what I'm intending to do with this presentation, to make one small contribution to overcoming those fallacies. We had those fallacies, I mentioned, lingual fallacies. Those fallacies which somehow force us. It is an ill intention of none of the participants in the debate. But people were trying to think, were forced to think by this lingual fallacy of a code as a thing which is separate, and interpretation as a thing which is separate. Two separate things, how to bring them together. But if you think differently, if you think in terms of never-ending process, where different types of processes interlap and evolve, one produce another one. You could see the processes of coding and the processes of interpreting as interrelated, continual, common stream of changes. And even before that, there's coming a simpler type of process, which are copying, multiplying, copying, just making new copies. Like elementary particles dispersing and multiplying in space and time. Very, very, very simple, elementary, not even physics, but something like a caricature of physics. Yeah. So this is the intention of this very presentation. Another whim of mine is I'm preoccupied with extremes. When I see this very complicated world of changing phenomena, I feel taken aback and I feel shocked because I'm disoriented. I can't understand it. And to be able to understand it, I'm looking for something very general and all-encompassing, and something very particular. I'm looking for extremes, because extremes are something like, well, why I'm standing here? Why I'm standing here? Because of the gravitation. I can jump, but I remain standing because there's a support. There's a limit where I cannot move further on. It's a limit. So I'm looking for limits. And when I'm looking for limits, I'm trying to see abstract. They are created by myself. They are created in my mind, and they are created in my lingual practices, which are called languaging. Yesterday, it was discussed already, languaging. I'm a great proponent of languaging revolution in linguistics. I'm cooperating with my colleagues in linguistics who are insisting that language as such is just an abstraction shared by linguists only. That people here around in this world, people at the railway station, or people in the hotel, and the people in the street, they do not practice languages. They don't have language. They are just languaging. They're just languaging. And they are using something which linguists later on interpreted as grammar, interpreted as vocabulary, interpreted as something this and that. Vocabulary as such never exists. Grammar is that natural. It's just a very helpful convention to interpret languaging, which is a never-ending cloud of practices, of people trying to communicate and being successful, miraculously successful. Why? Because of grammar, vocabulary, and some other things. So I'm extremely interested in big and complex, and simple and small. And another thing, and this is a slide from my 2-year presentation done 2 years ago in Alamode's simplest complex transformations. In my center in Moscow, we were practicing for about a decade, a dozen of years or something, this practice of simplest complex transformations, trying to cover the bridge between the most exquisite. I used to be the head of the Center for Advanced Methods in Social Sciences and Humanities. I resigned a couple of months ago because I just have no possibility to run it anymore for physical reasons and too overwhelmed by other obligations. But we were practicing, and still practicing actually. Informally, this center continues to exist. Transitions from the most elaborate methods in social sciences and humanities, and not only in social sciences and humanities. Actually, we were studying what they call transdisciplinary methodologies. Transdisciplinary, not interdisciplinary. Transdisciplinary are those methods or those practices which could be applied to all the disciplines. That couldn't be excluded, but could be applied to all, from mathematics to biology, from chemistry to psychology, and so on and so forth. And we wanted to reduce or to simplify. It's not actually, simplification is not reduction. We're not throwing away, we're just developing an equivalent but simpler versions. Purified, simplified versions of methods, trying to come to the other end. And the other end are elementary cognitive abilities. So I'm not going to tell more about this because it's just explanation why I'm having all my further things. So I'll be speaking about modes, about modifications, which are versions of modes. I'll be speaking about modality, which are specific qualities produced by those modifications. I'll be speaking about modules, which are small copies or small constituent parts, not copies, but small constituent parts which are copies. They are simultaneously both copies and constituent parts of certain modes. I'll be speaking about modal. Modal is something which is our instrument, which we are using, which we are making artificially. And I'll be speaking about modeling, which is this all-encompassing process which is equal to information, which is equal to, or informing. Modeling is, and informing, same things. So here's explanation of this intention. And to make it simpler, I'm using this, or we're using this as, and this is eigenform, so-called eigenform. A very simple thing, which with the help of eigenform, you can produce all kind of fractal structures. And here's a picture of universe or anthropic principle. We are here. When I'm saying we, that's not only humans. These are viruses and bacteria, plus all other creatures, plus ourselves, plus our biosphere. So we are kind of a departure point, and here we are going out there. We go out and over to a universe, and here we are going to in and down there into the elementary substances, particles, and so on. It's a well-known thing. So folding modes. Folding. I'd like to say that folding is a very interesting phenomenon. People here, of course, know about protein folding. But it's not only proteins that could be folded. So anything which is happening and which is becoming more and more complicated, but still integral, it has to be folded. And what is sequences, long sequences, you know, like a protein. You have a long string of signs, actually not signs, but of some elements. And you can read it. But actually to operate it, you shouldn't use it as a string, because it's too gross, it's too difficult job. You fold it to make it operation. When we are having this conference, it takes time, and there are too many people, and we are all together. And how we can put all those experiences, there are 2 or 3 dozen of people here sitting in this room all this time, and each having her or his specific perceptions, ideas, reactions, and so on and so forth. And to put them all in common, we have to fold them. And Marcella is intending beautifully to put this all, not all, but many parts, major parts, into a special issue of a journal. To fold it. So folding would be this journal, it would be further communication. This folding would be, there would be many foldings. I'll have my own story of what happened here, and I'll report it to my friends, and it will be my folding. This journal would be Marcella's folding. And each of you would have your own singular, or probably alternative foldings. Probably you'll have mixed impressions. And you can fold it this way, or you could fold it that way. So we have many foldings. So from this point of view, I think, and I mentioned here, Lyudmila Latskova, who hosted our previous 2 years ago conference in Olomouc. Well, a few years ago, about half a dozen years ago, she produced a PhD, a beautiful PhD, and a number of publications. And Lukasz, who is sitting here, he is her colleague and occasional co-author. so he can confirm this. That's a very essential contribution. She was presenting this PhD as a linguist, addressing protein folding, showing to linguists, and I told you that in linguistics now is in crisis. It has to jump away of this trap of fallacies, of separate grammar, separate vocabulary, separate this and that, into an integral folding-like structure. And Ludmila did this job. And yesterday, yes, I was very much impressed by Marcella's presentation, where she was presenting us with all those patternings, with all those extremely exquisite, complicated shapings of morphogenesis structures and so on and so forth. So I was really fascinated by her presentation, and I thought that that's very inspiring to me, to my own research. How you could interpret, and many of the things which Marcella wrote yesterday are running in great consonance with what I'm trying to. I could interpret Marcella's, and I'm sure that what I'm presenting now, she would give an alternative interpretation, and I would be very grateful for your comments in this way. So networking, patterning. By the way, also yesterday, so I'm not sure who did it, but somebody mentioned epigenetic landscapes by Conrad Waddington. So, you know, this modern synthesis provided us with a grand achievement of bringing together evolutionary theory with genetics. And this synthesis is really a tremendous achievement. But it was Conrad Eddington who immediately, at this stage, said that, no, there's something epigenetic, there's something additional, and he called this epigenetic landscape. And it was presented yesterday. And even today, it has been, mentioned that we have to look for, well, I'm very sorry that Robert Prince is not present here, because his 200-something codes, which is a great achievement of our code biology. Yeah? I think that's a very important thing, which really places code biology as a kind of one of the fundamental achievements in sciences general. So those 200-something codes are something additional. What happens with this basic code? But this basic code was multiplied. There were added some more things. But they cannot be, they're not something separate. They're something which develops over and out of genetic code. They're epigenetic in this, but they're so different, and they give additional possibilities for the, and actually when we're having all this epigenetic coding multiplying, we are having information and we're having meaning, like Branca mentioned. Meaning, there was a question, a legitimate question. Where is the meaning with elementary particles? Where is the meaning with elementary particles and elementary? The meaning is there. But the answer was given more than 10 years ago. It wasn't an answer, actually. It was a hint. A hint was given how to understand it, how to interpret it. In 2011, by Terence Deacon, in his book on incomplete nature, where he introduced the idea of absence, and where he introduced the idea of intentionality. We are all fallaciously guided by our own intentions and our own intentional abilities. Of course, we are intending something, but it was Terence Deacon who said that on the other side of the scale, development, evolution, genesis was able to occur only because something was missing. There was some stochastic structure, probably not fully stochastic, but largely stochastic structure, which was not complete, in the sense that not all the possible, well, you are calculating all the possibilities mathematically, and all the possibilities, they seem to be equal. No, of course, no, potentially equal. When you start calculating, your point of departure is that you calculate everything possible, and to the okay. It's a different debate. I'm a very simple guy who is simplifying things, and I'm always criticized by root simplifications. So that's a root simplification that we can think as this. I'm finishing up as stochastic. It's not stochastic because some things happened already, and as soon as they happened, they somehow already the noise is disappearing because something happened already. In this noise, some sounds already are being heard. In this stochastic structure where everything supposedly is equal is not equal. And this, what is not equal, actually creates what Terence called absence. And this absence is also creating not intentionality, but intentionality with E. Not I, but E. Intentionality. Intentionality is a driving force to bring about more probable, more happier combinations, happier constructions, happier processes, likely process, and so on and so on, and it goes on. And from this point of view, meaning and interpretation is present there, but it's not present there as a meaning we know, as a meaning we all experience, but as an absence of meaning which intentionally demand that anything what happens is also seen and interpreted from the point of a missing meaning. So from this point of, and I'm finishing up. So I took too much time of yours, and I'm finishing up, and I skip all my further slides. I'm only saying that what happens then? It happens that coding, which is actually a very exquisite variation of copying, very special. It produces a very exquisite and subtle type of coding, which could be seen as interpretation from the other side. Coding itself is not producing interpretation in its fine and clear form, but it produces such epigenetic types of coding which are nothing but in actual fact interpretations of the preceding. So from this point of view, there's no limit, there's no gap which you have to jump over. It's something like a bridge, it's something like an interface where earlier forms and processes intermingle with later, if we could speak about earlier and later, in inverted commas, of course. And where they are intermingled, they are folding. And they are creating interfaces. And those interfaces with us, and that's the last point for this linguistic fallacy, they could be interpreted as nouns. They could interpret it as something stable, as an initial moment and the outcome of a process. Those things could be nominalized. But anything which is happening in between are not nominal phenomena, but with their dynamic phenomena, their processes. Thank you. Sorry for taking too much time.
No, the fact is that we have a train and we have to finish on time. So I think that we have no time for discussion now because we are really in delay. But I apologize, but I asked to Fioretta to present the talk. We have to close on time this session. We can discuss tomorrow eventually about this interesting talk, but I have no solution in this moment.
Yeah, sure.
I'm sorry.
I shall also try to not to be too long. Okay. Now it is there. First of all, thank you for giving me a possibility to speak to this audience. I've been following code biologies since several years, but I am not a biologist, sorry, so I'm quite ignorant about biological processes. I hope I have something interesting for you to tell. It is about a theory that I did not contribute developing, or just in a very minimal part, but that which I'm trying to diffuse. It's called evidence theory. It is a mathematical theory of uncertain reasoning that is quite different from probability theory. Probability theory has a reference situation, which are games of chance. You throw dice. And when you throw a die, you know the set of the possible outcomes, the numbers 1 to 6. So it is a closed world. In a closed world, the meaning of each outcome can be taken as given. So in probability theory, you can forget about meaning. You use a concept of information that, unlike yours, does not include meaning. Shannon's information theory is based on probability theory. Even the act of throwing a die, you can interpret it in terms of Shannon's information theory, like you have an initial probability of each phase, and then your die may hit this bottle and then that glass, and those are noise on this channel that finally generates what phase comes out. Shannon's information theory may be appropriate for a purely extremist neo-Darwinian theory of evolution, where mappings between genotypes and phenotypes, between codons and proteins, insofar I understand, are occasionally disturbed by random mutations. The reference situation of evidence theory are either a judge evaluating testimonies or a detective evaluating clues. Now, even if you are not a reader of detective stories, you know that in detective stories, at the very end, a denouement takes place. Oh, things are so different from what I've been reading up to this point. novel possibilities enter the picture. It is an open world. What is important to do in an open world, not staying at prima facie, what information comes first, that's what Dr. Watson does, but actively looking for cues, reinterpreting cues. Clue can be interpreted in several ways, and henceforth I shall make a distinction between organic codes and cultural codes. I'm not sure that the distinction I'm going to make is the one is the correct one. I may be making mistakes, but I'll tell you later on where I see the discrimination, where I've put it. If it is if you don't look for cues, if you limit yourself to what is arriving at your available, you may have different interpretations. So Dr. Watson has one interpretation, but there might be several Watsons coming to different interpretations. All of those interpretations would be generated without thinking. And I think that roughly what might happen in what we eventually call organic codes in biological living beings that have no ability to make predictions. But unlike Dr. Watson, Sherlock Holmes looks for cues. He formulates hypotheses out of predictions that he eventually makes out of a few cues, maybe out of abduction. It's a cultural code. A bit of mathematical framework. A testimony in evidence theory is a set of masses that are relative to possibilities. So here, A1, A2, A, and A are possibilities, and M are the strength of the evidence supporting each possibility. Pay attention, unlike probability theory, these possibilities are not mutually exclusive. When you throw a die, you either have phase 1 or you get phase 2 or 3 or 4. Whereas in this case, when you have different testimonies, well, they may say things that partially overlap. So the possibilities that are used by evidence theories are sets. And evidence theory sees probability theory as being based on singletons, sets that entail one element only, phase 1, phase 2, phase 3. What happens with singletons? They can be either distinct or overlap, whereas the possibilities, the testimonies in evidence theory may partially intersect. Second testimony arrives on possibilities B1, B2, B, and B, and of course it may happen that the possibilities envisaged by the second testimonies partially overlap, intersect those of the first testimony. They may not have a void set. This is not pure maths. This is more used by engineers than by mathematicians. And one typical application is in data fusion, which means, for instance, combining different databases or combining information from different sensors in order to distinguish enemy missiles from other flying objects. Unfortunately, increasingly common application. Now, one very tentative bridge I'm trying to throw is I've read that you are eventually concerned with statistical proteins and what might have happened in the early stages of lives when mappings between codons and proteins was not very clear. That might have been some intersect. Maybe evidence theory could be useful in order to understand the formation of codes. That is, you receive certain proteins one time, another time you provide a feedback, and somehow a different, slightly different coding appears. So in probability theory, you can have only discrete jumps, whereas here you can have possibilities that more intersect to a greater or lesser extent out of feedbacks. No, this was the possibility set of evidence theory is called frame of discernment. It is not just a different name. Of course, it conveys the ideas that cognition is important, but there is a technical difference. In probability theory, the possibility set is an algebra. What does it mean? It is a set of objects plus operators on it. In evidence theory, the frame of discernment is simply a set of objects. No operator is defined. Let me explain with a stupid example. We have been enjoying a nice buffet at the restaurants. The buffet has a set of pastry and coffee and cappuccino, and apparently the Italians have a rule that they cannot drink cappuccino in the afternoon. So for Italians, the buffet is an algebra. But although I am Italian, I do enjoy cappuccino in the afternoon. So for me, the buffet is not an algebra. Why is it so important? Because in probability theory, it is assumed that the possibility set is a sigma algebra, which is an algebra where the complementation operation has a paramount role. What does it mean, complementation? It means that you can define a residual event. So you think of certain possibilities, you make a list, and then if you fear that your list is not exhaustive, then you define a residual event like anything else that might happen. In this way, you reduce any problem, any decision problem you face in life to the problem of throwing a die. A die has a given number of faces, and if you use a complementation operator, you can define a given number of possibilities. That's a world. You transform an open world into a closed world. That's why probabilists say that you can apply probability theory everywhere, which is why probabilists hate evidence theory. But engineers like me use it. Since the frame of discernment is not an algebra, it is also possible to assign a mass of evidence to the whole set, to the whole probability set, without this mass going down to the single subsets. This mass is hovering the frame of discernment. It does not pertain single possibilities. What does it represent? Well, it can represent uncertainty on what I know about these possibilities. Can probability theory account for this sort of uncertainty? Yes, it can. There is an extension which is based on lower and upper probabilities. The most important form are the lower probabilities, which are also eventually called the sub-additive probabilities. So these probabilities do not sum up to 1. If you are playing dice in a clandestine place, you may fear that the die is not fair, in which case you may say, well, let me start with assigning a probability 1/10 to each phase. Clearly, the sum is less than 1. These are sub-additive probabilities. You restrain judgment. So you can account for this with a suitable extension of probability theory. But still, you cannot account for partially intersecting possibilities, and you cannot account for the fear that something happens that you are not even able to figure out. So what is the difference between Dr. Watson and Sherlock Holmes? Why does Dr. Watson stop at the first set of possibilities he's seeing? Because to him, if there is any sort of contradiction, well, maybe that testimony was drunk, maybe Dr. Watson ignores certain details that, by contrast, ring a bell in Sherlock Holmes' mind. Sherlock Holmes' suspicion of contradiction. He starts thinking, here there's something I should investigate. Starts out investigation. Maybe for most of the book, most of the detective stories, Sherlock Holmes looks like a crazy guy, but at the end, at the denouement, it appears that Sherlock Holmes is able to say, poor Watson, that detail was so important. So, can we represent this within evidence theory? Yes, within one special branch of evidence theory that has been developed in part by Philip Smetz, which enables you to assign a positive mass to the void set. What does it mean? You attach a mass to something that is void. It means that you feared that something else might be the case, something that is not within the current frame of discernment. And I've read in one of your publications that cultural codes change very quickly. I may tentatively interpret that in the sense that in cultural codes, M of the empty set is very large. So you see lots of contradiction, you change your interpretations very, very often. Once again, a bit of math. Evidence theory is very much about the combination of evidence. In a closed world, so if you know the list of possibilities, and still you have you retain the freedom of assigning a mass to theta. If you receive two testimonies, A and B, you combine them by means of what is here called the Dempster-Shafer combination rule. It is very, just grasp the meaning. On the numerator, you find what is common. So the intersections between the composing possibilities. What is common between the two testimonies? And the product of the weights of these two testimonies. At the denominator, you have 1 minus what the extent to which these two testimonies do not intersect. So the extent to which they are conflicting testimonies. So Dr. Watson takes this conflicting mass and by subtracting it from 1, redistributes it to the existing possibilities. So you have the X. And by the way, in the special case where these possibilities are singletons, Dempster-Shafer combination rules boils down to Bayes' theorem. If you are in an open world, then you eventually use Smet's rule, which is just the numerator. You don't use the denominator because you do not redistribute conflicting evidence among the acceptable possibilities. In which case,
a renormalization is absolutely necessary, including M of the empty set. In the case of the Dempster-Scheffer rule, you can use renormalization encompassing M theta, the frame of discernment, but not M of the empty set, but it's not strictly necessary for the theory. Also because since these possibilities can intersect, you don't have possibilities summing up to one. It doesn't have the same meaning. Now, suppose that you have a living being who's sufficiently sophisticated to make predictions about the future. By means of these predictions, this living being may be able to make hypotheses. This is what I tentatively called cultural codes. I'm not sure about the names. Because I read that you've been studying stories about a wolf continuing to chasing a prey behind the tree, whereas the snake stops. So in this usage of this expression, a wolf has a cultural code, whereas a snake does not. But I'm not a biologist. I may be making huge mistakes. Please correct me. So these sophisticated living beings makes hypotheses and eventually tests them. In evidence theory, you have two measures on hypothesis. This is called the belief functions and the plausibility function. The belief function simply takes all the evidence. Look here at inclusion, that is included in your hypothesis, whereas the plausibility function takes any evidence that intersects your hypothesis. Obviously, plausibility is generally larger than belief. Now, it is very important to understand that in evidence theory does not specify how you formulate hypotheses. You may formulate hypotheses, you, the user of evidence theory, on a subset of the frame of discernment, where you may be free to define any logical operator, including complementation. But you're not forced to do that. You may formulate hypotheses out of divine illumination. It is absolutely fine. It is not specified by the theory. Why is it so important? Because Chomsky, our common enemy, says that there are logical rules that explain any language. Classical artificial intelligence of the 1950s used to say that intelligence is logical rules. Probability theory also says there are logical operators on the possibility set, whereas evidence theory says, well, with evidence theory splits what probability theory sees as one single process in two phases. The first phase is receiving information within your frame of discernment. No operator. The second step, if you are able to make prediction, then you may make hypothesis, and if you want, you may be a logical person and make your think your hypothesis by means of some logic. But those steps are separate. Step one, step two. So please don't be scared by the maths. It's just that you consider all the evidence supporting your hypothesis, or also the evidence that is partially supporting your hypothesis. So evidence theory is not telling you how you have to think. Evidence theory is simply telling you how you should sensibly, rationally combine the empirical evidence you receive. The way you think does not pertain to evidence theory. It is more shy than probability theory. How does Sherlock Holmes figure out his hypothesis? Well, by zooming in and zooming out the frame of discernment. To the extent that Holmes goes into the details, he's tightening the frame of discernment. He goes back to the scene where the murder has taken place and asks neighbors looking for other clues. Oh, thank you. Whereas Watson coarsens the frame of discernment. Arthur Conan Doyle depicted Dr. Watson as a quintessence of stupidity, whereas Holmes is very intelligent, but this happens only in detective stories. In broader real life, coarsening the frame of discernment may be very useful indeed. So sometimes we need a bit of Watson. These two processes of tightening and coarsening the frame of discernment, they are both useful. The wrong taste. Has anyone seen that movie? Who has seen 12 Angry Men? A few. It's about a jury coming to a verdict that is opposite to the initial one. How do they do that? By constructing a coherent narrative. So here we come to a third level, which is, well, I don't have a name. It may be cultural code is the right name for this third level, but once you have several living beings who are capable of formulating hypotheses, then the combination of this hypothesis through a linguistic ability to communicate gives rise to a complex narrative, and that changes things at still a further level. There are models that could be used to expand on that. Constraint satisfaction network, I don't show you anything, but let me come back from cultural code to organic codes. Bacteria are able to switch between alternative macroscopic forms in reaction to environmental stimuli. So this is pictures I've seen of bacteria that may switch between these two macro states depending on the chemicals they are facing. Now, bacteria are certainly not able to figure out a prediction, to make prediction, but they are living in an open, unpredictable world because there exist enemies who are called researchers who are develop antibiotics, and they develop novel antibiotics. So bacteria must be able to interpret novel antibiotics in terms of partial intersection, similarity with antibiotics that they were previously acquainted with in order to develop a novel response. Coming back to the organic code, even without prediction, you can possibly need some bit of evidence theory. I think I'm really exhausted times, so let me stop here. The other slides were not so important. Thank you for your attention.
Time for a short discussion because I have to distribute the tickets and you need a lunch before to leave. Discussion? Someone? Yes, please.
It's just an observation. So Sherlock Holmes is not doing what he says that he's doing.
Sorry?
Sherlock Holmes is not doing what he says that he's doing and deducing the truth. Actually, he's doing something more like theory choice based on something like theoretical virtues, and which is more encompassing and which is simpler and which, right? So it's a totally different operation than what he claims that he's doing. You see how I totally missed all the biology and got the analogy?
Oh, I'm too ignorant about Sherlock Holmes.
Well, never mind, it doesn't matter.
So he's not... that's absolutely important. So when you are zooming in the frame of discernment, you're not doing deduction. Absolutely. You could zoom in several directions, and you do it by abduction. Absolutely. Yeah, or theory choice, which evidence theory does not specify. You're free to do to behave the way you want.
Okay, thank you very much. I have to close. And now I distribute the tickets personally. We have 2 tickets, one way and return. Make attention, please. Allora, tu prendi il ritorno. So,